we are continuing to explore trends in the development of animals that are useful in the classification of animals into the various taxonomic groups within the animal kingdom. And one of the things that I want to do as I begin to tell you more about developmental trends is review a little bit what we had previously learned concerning animals that have 3 layers of embryonic development. We also call these germ layers. So for animals that have 3 germ layers, which are the ectoderm, the endoderm, and the mesoderm, we are going to subdivide them into 3 groups. We know that there are those that have a true coelom. We call them the coelomates or eucoelomates. The prefix U, the letters E and U, means truly or true. And so in that case, will be animals with a true coelom. We also have animals that develop a cavity where organs can form, but that cavity is going to be known as a pseudocoelom. And we also have animals that don't have a cavity where organs are going to be forming. The space between ectoderm and endoderm is filled pretty much with mesoderm cells. And so those are going to be known as the acoelomates. And the main group of animals we're studying here are going to be those worms of the phylum Platyhelminthes. And so one of the things you may want to know a little more, so what exactly is the difference between a true coelom and a pseudocoelom? And so I wanted to explain that to you in this video. And I wanted to show you in this illustration. If you look at this space here, you can see that there is like an opening inside of this red layer for the animal. The red represents the mesoderm. And so if we could, like they do in some of those movies these days, where you can become so small, you can travel inside of an organism instead of a person. But in this case, we're going inside of the worm. If we could go in here into this space known as a true coelom, and you stood right here and you look in all directions, what you're going to see is only mesoderm. And so by definition, a true coelom is a cavity or space that is completely surrounded, completely formed by mesoderm cells. If we look at a pseudocoelom, and we could travel, again, made ourselves so small we could fit inside of a nematode worm, and we go into that space known as a pseudocoelom. If you look in the direction towards the skin, you're going to see that the cells here are of a type we call the mesoderm. But if you look in the direction of the gut, where food is passing through, those cells are going to be endoderm. And so we are finding ourselves in a space that is not completely made in a uniform fashion, meaning that on one part you can find endoderm, in another part you can find mesoderm cells. And so that is the main distinction between a true coelom and a pseudocoelom. Remember, true coelom is a space that is formed and completely surrounded by cells of the mesoderm. The reason I wanted to mention this to you is because as we continue exploring more of these developmental trends, I want to emphasize that protostomes and deuterostomes are subdivisions for animals we call eucoelomates, or animals that have a true coelom. One more detail that I wanted to mention before I move on to the next slide is that only animals with 3 germ layers are going to be designated as eucoelomates, pseudocoelomates, or acoelomates. Only animals that have 3 germ layers are going to be used for this designation. And so don't trick yourselves, or don't let anyone trick you if they ask you, hey, what about a jellyfish? What is a jellyfish? Is a jellyfish a eucoelomate, pseudocoelomate, or acoelomate? And the answer is none of those. The reason is that cnidarians, like jellyfish, only have 2 germ layers. They are diploblastic. So remember, only animals that have 3 germ layers or triploblastic will be the ones we designate as one of these 3 names we have just reviewed. But now we're about to focus only on animals that have a true coelom. and those are known as the eucoelomates. Eucoelomates, depending on the development of the mouth and other characteristics of the embryo, can be subdivided into protostomes or deuterostomes. And so what we're going to concentrate on first is going to be the protostomes. Protostomes are going to be animals that are going to form the mouth first. So let's take a look at this illustration of an embryo. And so here we see a gastrula stage that has all three germ layers: ectoderm in blue, endoderm in yellow, mesoderm in red. And so here we can see that there is going to be an opening known as the blastopore, and this opening leads into a second cavity that forms in the embryo, which is known as the archenteron. If you look at this first part of the word here, arc or arch, it really means what it says. This here is formed like an arch. What? Enteron. Enteron is the same as enteros, which means gut. So archenteron is going to be this arch-shaped gut. But what we're trying to focus on right now is going to be the entrance into this archenteron known as blastopore. So with animals we call protostomes, this first opening of the embryo into the archenteron becomes the mouth. Consequently, the word protostome, it has a literal meaning. Proto meaning first, and stome means mouth. And so the mouth forms first. That's what the protostomes are. Animals where during the embryo, this first opening, the blastopore, goes on to develop into the mouth. There are going to be other characteristics we must be able to recognize about protostomes, and one of those is going to be the pattern in which cells are cleaving. During cleavage, remember this is going to be the very first stage in embryonic development. During cleavage, when a cell divides first into 2 cells, then those 2 cells divide into 4 cells, and you have 8 and 16. Notice how the planes of cleavage are not going to be parallel or perpendicular to one another. So here we see a plane of cleavage that divided a cell into 4 down here in the bottom. The next cells that are forming at like a 45-degree angle in relationship to the previous plane of cleavage, which is right here. And so because of this, as the cells are added, there's going to be like a spiral pattern that is going to be emerging. That's called spiral cleavage. Another characteristic of the protostomes is that the coelom is going to be formed in a way that cells that began near the blastopore, keep in mind the blastopore is this opening into the archenteron, cells that used to be here near the blastopore are going to be emerging and eventually migrating, splitting into a space where the blastocele used to be. And so this ingression, by ingression I mean there's a migration inwards of these bands of mesoderm cells, is what we call splitting of the coelom. And there is a name that I want to write over here that will be useful for later reference. So when the coelom forms by splitting from the blastopore, these animals are going to be known as schizocoelous. I'm missing a U over here. Schizocoelous. Schizo means split, and coelous is making reference to the coelom. One last detail I want to tell you about the protostomes is what happens with regards to the embryos. The embryos are going to show what we call determinate development. This concept of determinate development means that if we were going to take one of these cells from this 8-cell embryo, if we were to take one out, you may ask, what would happen to that cell? Is it going to continue developing into an animal? And the answer is no. Removing a cell from an animal that shows determinate development means that that cell has already received enough information to eventually take an identity to be maybe part of the torso of the animal, part of the head of the animal, part of the tail of the animal. And so that cell is not going to develop into a complete embryo or eventually become an adult. Development is going to be terminated because it's not going to have all of the parts that the embryo or eventually the adult animal is going to need. What about the other 7 cells that remain? Those 7 cells will unlikely develop into an adult animal either. And so those cells are going to be missing that one that maybe was going to be forming part of the head or the torso. And so with that information missing, with that cell missing, both the 7-cell embryo and the 1 cell out of the 8 you removed will fail to develop. That is going to be what we call determinate development. Now, keep in mind, we're talking about animals that have a true coelom. The other alternative for animals with a true coelom would be those that we call the Deuterostomes. And these animals called the Deuterostomes have their own characteristics. Let's go once again and take a look at the opening into the archenteron, arch gut. This opening, called the blastopore, still the same name, that hasn't changed, will not be the mouth of the animal. For most animals, this is going to be the anus in the feeding canal. So where is the mouth? The mouth doesn't show here in this illustration. The mouth is going to be forming eventually later from an opening that doesn't ever receive a name. We call that just an unnamed opening. So notice what happens. The anus forms first in a deuterostome. Mouth forms last or second. And that is the literal meaning of deuterostome. Deutero comes from a root that means second. Stome means mouth. And again, putting the two together means that the mouth develops second to the anus form first. Other characteristics we can recognize of this deuterostome development is that cleavage is going to be radial. Radial cleavage means that every time cells are dividing during the cleavage stage of development, the planes of the cleavage furrow, the planes of cleavage are always going to be either parallel to the previous plane of division or perpendicular, meaning that it's going to be at a 90-degree angle. And so if this was the previous plane of cleavage, notice how the next one is going to be perpendicular to the previous one. And this plane here is going to be parallel to this plane over here. That's called radial cleavage. Other characteristics we should remark is that these animals are going to be forming a coelom from a band of cells that are going to be first part of the endoderm, part of the archenteron gut, and these are going to differentiate, meaning that they're going to become mesoderm as out pockets of this primitive gut present in the early stages of development for this gastrolab. So what would happen to these out pockets of the gut is that they will eventually fill a portion of this space here, what used to be the blastocele, and that is where they are going to be forming a complete coelom or true coelom. So this type of coelom development that comes from the gut, remember what I said before, entero means gut, also has its own name that I wanted to mention right now. And basically, it's here. They have enterocoeli. So the animals are known as enterocoelous. The animals are going to be known as enterocoelous. Why? The coelom developed from the gut. I'm sorry about that. And no, wait, let me take a break here. Okay, I'm sorry about that. I am back. And the last detail that I wanted to tell you about the Deuterostomes, animals where the mouth forms second, is that the cells that are forming during the embryo, they are going to show a different type of development that we call indeterminate development. And this indeterminate development means that if we were to remove one cell from this 8-cell embryo, what would happen to that one cell and the other 7 cells that remain together? Both parts will continue to develop as if nothing happened. The one cell you remove will cleave and become 2, and then 4, and then 8, and 16. And the 7 cells over here will continue their pathway of development through cleavage into the blastula, into the gastrula. Both parts will develop into a complete embryo and eventually into a complete adult animal. And so that is what we mean by indeterminate development. Each cell has the potential to become an entire complete organism eventually if development is allowed to continue. And so that is all that I have for you with regards to patterns of development and specifically what has to do with Protostome versus the Deuterostome animals, which are the subdivisions for animals that have a true coelom. I will have one more video on body plans, development, and classification for you.